Most of the calcium in the body is stored in the bones, where it can be mobilized if necessary. In the cytoplasm, calcium concentration is very low since it activates several cellular processes, such as muscle contraction and the release of neurotransmitters. The main sources of calcium in the diet are milk and its derivatives. Moreover, smaller amounts of calcium can also be found in fish, cereals, and legumes. When the diet is rich in calcium, its absorption in the intestine occurs primarily through a paracellular pathway, crossing the tight junctions located between two adjacent cells of the intestinal mucosa. This process takes place in both the small and large intestine by passive diffusion, because calcium moves down its concentration gradient. When the intestine contains a low calcium concentration, this is mainly absorbed in the enterocytes of the duodenum and proximal jejunum. This absorption is carried out by a transcellular pathway which involves active transport. Calcium enters the enterocyte from the intestinal lumen by selective channels TRPV6 in the apical membrane through a passive diffusion process. Calcium moves down its electrochemical gradient by passive transport due to the low calcium concentration in the cytoplasm and to the attraction by negative charges located inside the plasma membrane. Calcium binds to the cytoplasmic protein calbindin, which transports the cation to the basolateral side of the enterocyte. Calcium can also be introduced inside endoplasmic reticulum vesicles. All these processes avoid the presence of high concentrations of calcium in the cytoplasm, which would be cytotoxic. Calcium leaves the enterocyte and moves to the interstitial space by crossing the basolateral membrane against its concentration gradient, for which energy is required. Calcium binds to a calcium ATPase placed in the basolateral membrane, which hydrolyzes an ATP molecule and uses an energy release to propel the calcium outside the cell to the interstitial space. To a much lesser extent, calcium can also cross the basolateral membrane by way of a calcium-sodium exchanger. In this case, 3 sodium ions enter the cell down its concentration gradient, which forces the exit of a calcium ion to the interstitial space.